So, good morning. Today we'll be seeing the next topic from genetics, which is a small topic, which is a deviation from Mendel's postulate of dominance. And the name of the topic is incomplete dominance. Okay, so what we are going to see today is from incomplete dominance. So, today we are going to see about incomplete dominance. Now, incomplete dominance is also called as or is also termed as phenotypic blending. It is also termed as phenotypic blending. Incomplete dominance is basically a monogenic, which means single gene, monogenic, quantitative inheritance pattern. Monogenic quantitative inheritance pattern. Now, if you look at what incomplete dominance is all about, incomplete dominance was first actually observed by a scientist called Carl Correns. We will see in detail a scientist called as Carl Correns. He actually identified or observed incomplete dominance. And Carl Correns has observed incomplete dominance in a plant which is called as the 4 o'clock plant, and the scientific name of this 4 o'clock plant is Mirabilis jalapa. Mirabilis jalapa. In Mirabilis jalapa, Carl Correns has studied this or identified this incomplete dominance. Okay, so this is one example, and it is a very common example. Along with this Mirabilis jalapa, incomplete dominance is also seen in the flower color. It is also seen in the flower color. Carl Correns also studied 4 o'clock plant Mirabilis jalapa flower color only, where he has identified the incomplete dominance. It is also seen in the flower color of a plant called snapdragon. And the scientific name of this plant is Antirrhinum majus. Antirrhinum majus, also called as dog flower or dog plant. It is called as a dog flower because when the flower is opening, the opening of the flower seems like the opening of the mouth of the dog, so it is called as dog flower. So in these two cases we identify incomplete dominance. So we will see incomplete dominance and we'll try to understand what incomplete dominance is using the flower color of Mirabilis jalapa. Okay, so Mirabilis jalapa, if you're looking at the flower color of Mirabilis jalapa, red color flower is dominant. So the phenotype of the red color flower, this is the phenotype. Red color flower is the dominant phenotype, and white color flower is the recessive phenotype. Okay, so when you're taking red color flower, the genotype of the red color flower is homozygous dominant, capital R, and white color is small r, which is homozygous recessive. So what is the gamete that is formed from here? The gamete that is formed from here is going to be capital R. Only one type of gamete is formed because it is homozygous condition. So here also you will have only one type of gamete that is formed because this is also what condition? Homozygous condition. So this is your parent, this is your parental generation, parent. This is your what generation? Sorry, this is your gametes. This is your gamete. And fusion of the gamete will result in the formation of the F1 hybrid, capital R, small r, F1. Now, from here understand it very carefully. If it was Mendelian, if it were Mendelian crosses, when we have studied Mendelian monohybrid crosses, we have always seen that the F1 progeny or the F1 hybrid had either of the parental phenotype. Either of the parental phenotype, usually it would be what? Dominant in the F1 in Mendelian crosses. Okay, now, but here what happened is, to the surprise of Carl Correns, in the F1 generation, he identified a third phenotype which is not resembling any of the parent, and the third phenotype that he has observed in the color of the flower, that is pink. So what did he identify? He has identified pink color flower after crossing a homozygous red color flower plant with a white color recessive plant. F1 generation showed a very different phenotype, which is not obeying the law of dominance that was proposed by Mendel. So what he identified here is, the plant is showing what color? Pink color. No, sorry, the flower is showing what color? Pink color. So this pink color flower is an intermediate phenotype. This pink color flower is an intermediate phenotype. This pink color flower is an intermediate phenotype. It is not resembling any of these two parents. Okay, the flower color is different here. Now, what is the reason for having a different pink color flower? So, when Carl Correns was actually analyzing what is the reason for having a different color flower here, or an intermediate phenotype which is not resembling either of the two parents, what he actually thought is, or what he actually proposed is, the dominant allele here, which is the capital R allele, the dominant allele is not completely suppressing the expression of the recessive allele. To understand it, according to the NCERT, the statements of the NCERT, this dominant allele, which is capital R, is not completely suppressing the expression of what? The small r, or the dominant allele is not completely dominant over. I can write it like this: the dominant allele that is capital R, the dominant allele capital R is not completely dominant over, it is not completely dominant over the recessive allele small r, as a result of which we are getting what? An intermediate phenotype, which is capital R, small r genotype. The intermediate phenotype is pink. Now pay attention here very carefully. Now what we should understand here is, of course, this is what it is actually given there in the NCERT. The dominant allele is not completely dominant over the recessive allele, but if you see, because it is not completely dominating, suppressing the expression of the recessive allele, or because it is not completely dominant over the recessive allele, we are calling it as what dominance? Incomplete dominance. Now, I have also said that this is monogenic quantitative inheritance. Now, what is the meaning of monogenic quantitative inheritance? First, understand what is the meaning of this word quantitative inheritance. Quantitative inheritance means, we'll see to the small example. Now let us take, for example, we are having something like capital T and capital T, the Mendelian capital T, where capital T is giving the tallness of the plant. And let us take capital T, both dominant alleles are producing 10 centimeters height in the plant. Okay? Now we have one more condition where there is capital T, small t. This is also producing a tall phenotype, and this is also producing 10 centimeters height in the plant. The third case is, let us assume we are having small t, and we know that this plant is what plant? Dwarf plant, and this is producing what? 5 centimeters height. This is a recessive trait, recessive character, or recessive expression of the gene. Now here there are two dominant alleles. Still there is an expression of 10 centimeters height. Here there is a single dominant allele. Though there is a single dominant allele, still there is expression of what centimeter? 10 centimeters height. Means, in this case, the number of dominant alleles are not playing the role in expression of the dominant phenotype. Whether there is a dominant allele or no is what is important here to show what expression? The dominant phenotypic expression. So in this case where we are talking about the height of the plant, number of dominant alleles are not playing an important role in expressing the phenotypic dominant trait. But what is important is whether there is a presence of the dominant allele or no. On this kind of inheritance pattern where this is quantitative, this is qualitative. So this kind of inheritance pattern where number of dominant alleles don't play a significant role, but what plays significant role is whether there is a dominant allele or no. So this kind of inheritance pattern is qualitative inheritance. Now for example, the height of the plant in the case of Mendel's crosses with pea plant, all of them are qualitative traits. So if you look at quantitative trait, let us take for example, there is capital R, and this capital R, let us take it is producing probably some 10 micrograms of red color pigment in the flower. Just hypothetical example. Capital R, small r is producing, let us take only 5 micrograms of the pigment. 5 micrograms of the pigment. Let us take small r is not producing any red color pigment. Is not producing any pigment. So now if you look at this, two alleles are present here, two dominant alleles are present here, and when both dominant alleles are present, we are getting 10 micrograms of the pigment. When there is only a single dominant allele, we are getting how much? 5 micrograms of the pigment. Now, if you see the difference here, each allele is contributing to 5 micrograms. When there are two dominant alleles, 5 micrograms plus 5 micrograms, we are getting 10 micrograms. But when there is only one dominant allele, the amount of pigment, the amount of red colored pigment that is produced has decreased by 50%. Correct? So here the inheritance pattern or the expression of the phenotype, or what amount of phenotype or what is the intensity of the phenotype that is expressed, is also depending on what? It is also depending upon the number of dominant alleles. So if there is only one dominant allele, amount of pigment formed is less. If there are two dominant alleles, amount of pigment formed is more. So in the case of quantitative traits or quantitative traits depend upon the number of what? The number of dominant alleles that are present. So the flower color that we are seeing here, it is what kind of inheritance pattern? It is a monogenic quantitative kind of inheritance where the number of dominant alleles play a significant role. So if you are seeing this Mirabilis jalapa 4 o'clock plant also, I have written it as monogenic quantitative inheritance because there is a single gene that is involved here, and it is a quantitative trait. Presence of one dominant allele or two dominant allele is changing the phenotype of the color of the flower. Okay, so that is why it is called as monogenic quantitative inheritance. Next one is phenotypic blending. Here actually it is not a blending pattern of inheritance, but many a times we say that it is phenotypic blending, assuming that or taking into consideration that there is an intermediate phenotype that is produced here, assuming that the phenotypes have got blended, but in real it is not phenotypic blending, because as we see in the further generations we will get to know that it is not phenotypic blending. So I think it is so far clear till here. What we are talking about is incomplete dominance, point number 1. So incomplete dominance is the dominant allele is not completely dominant over the recessive allele. As a result, it is producing what? An intermediate phenotype. But as we have discussed just now, that now let us, to understand it much better, let us take, there are two alleles, and these two alleles are capital R and capital R, and this two allele is capital R is producing some amount of pigment, this capital R is producing some amount of pigment, and let us take this amount of pigment is giving you red color. And let us take, for example, there is a capital R and a small r. Capital R can produce pigment, but the small r cannot produce pigment because it is recessive trait. As a result of which we are only getting what? An enough amount of red color pigment is not present. As a result, we are getting what? Pink color of the flower, because only 50% of what is produced when there are two dominant alleles is produced here. Now let us take small r, there is no pigment that is produced here. because the gene is not getting, the small r gene is not producing any pigment. The small r gene is not producing any pigment, so the amount of pigment that is produced is how much? There is no pigment here, so that's why there is no color here. The flower color is what color, beta? White color. Okay, now let us move forward. Let us move forward. Now if you are crossing this F1 hybrid, if you are selfing this F1 hybrid, what do we get here? Two gametes will be getting capital R, small r, and here also you will be getting capital R and small r. So if you take a Punnett square, if we make a Punnett square, here capital R, small r, the gametes I am writing here, and these gametes are here. So capital R, capital R, capital R, small r, capital R, small r, and small r, small r. So this capital R is going to give you red, this is going to be pink, this is also going to be pink, and this is going to be what? White. So if you look at the phenotypic ratio, if you are looking at the phenotypic ratio is 1:2:1, where number of red is 1 out of 4, number of pink is 2 out of 4, and number of white is 1 out of 4. So this is a little deviation from the Mendel's law or Mendel's monohybrid crosses. Mendel's monohybrid crosses were showing complete dominance, so we were getting a ratio of what? 3:1. But here in incomplete dominance, the phenotypic ratio that we are getting is what? 1:2:1. So this is actually incomplete dominance is also a deviation from, it is also a deviation from what? From Mendel's postulate of, it is also a deviation from Mendel's postulate of dominance. Mendel's postulate of dominance. It is a deviation. Okay, expected one is 3:1, but we did not get 3:1, we got what? 1:2:1. If you look at the genotypic ratio is also 1:2:1, capital R is 1, capital R, small r are 2, and small r is 1. So 1:2:1. So here in the case of this monohybrid cross, if we see the phenotypic ratio and the genotypic ratio, both of them are same. Both of them are 1:2:1. This is also 1:2:1. So this is about the monohybrid cross, which is a deviation from the Mendel's law, which is also what? Explaining the incomplete dominance. So the cross that we did here is a monohybrid cross, but it is deviating from Mendel's law of or Mendel's postulate of dominance. Actually we should be getting 3:1, but we are getting 1:2:1, which is telling you that the expression of the dominance of the capital R over the small r is not complete. Okay, but we know that it is not the phenotypic blending, or it is not phenotypic blending, because it is a quantitative trait. Capital R is producing some amount of protein and small r is not producing any amount of protein, so that's the reason we are getting intermediate phenotype. Okay, so we will see one more example which is given in the NCERT about this incomplete dominance. The second example that we'll be seeing is the starch grain size. Okay, starch grain size in the pea seeds. The next example that we'll be seeing is the starch grain size. Starch grain size. Okay. So in the case of pea seeds, there are seeds which have large starch grain, and a plant with large starch grain containing seeds is crossed with a plant which is having small starch grain. Let us take for example, this is a seed and the starch grains are large starch grains. See the starch grains, they are large. So this large starch grain is represented with capital B, capital B. Here we are having small starch grains represented with small b, small b. So the regular cross, monohybrid cross, small b are the gametes, cross both of them, capital B, small b, this is F1 generation. Now if it is a Mendelian cross, a typical dominance case, here we should be getting large sized grains, but here instead of getting large sized grains, we have got starch grains which are of intermediate size. Okay, we have got what size? Intermediate size starch grains we have got. Okay, so it is neither large nor small, so we have got intermediate size starch grains. So F1 hybrid is showing an intermediate phenotype of both the parents. Okay, so again we do a selfing here with the same F1 hybrid. So we will be getting capital B, capital B, capital B, small b, capital B, small b, and small b in 1:2, 2:1 ratio. Both phenotypically as well as genotypically we will be getting the same ratio, 1:2, 2:1. So this is going to be having a large size starch grain, and here the starch grain is going to be of intermediate size, and here the starch grain is going to be what? Small. Okay, so this is about the incomplete dominance. Okay, thank you very much.